It's hard to believe it, but it's already week 2 of Bio 304. And you don't have a test yet due this last Sunday, but test number 1 and test number 2 will both be due this coming up Sunday. And the structure of the tests are going to be, you've got 10 multiple choice questions, and then you're going to have 5 essay questions. And 4 of those essay questions are going to be worth 5 points, but one of them is going to be worth 10 points. And as we study this material, I want you to think about how well you understand it and the level and depth of knowledge. So for a 10-point essay question, I'm expecting quite a bit. Let me give you an example here. This week, we're going to talk about photosynthesis and transport in plants. So transport, we're going to talk about both the xylem and the phloem. And of course, the xylem is going to move water from the roots to the leaves, and then the phloem is going to move secondary metabolites. Secondary metabolites. That's a fancy way of saying organic molecules made after the carbohydrates in photosynthesis. So like the green pigment chlorophyll would be a secondary metabolite. Okay, and then we're going to study how the phloem moves the secondary metabolites from the area where they're made, mostly the leaves, and transport them through the rest of the plant. Okay, now as you study this material and you're going to get these essay questions, think about the difference between superficial knowledge and really deep understanding of the material. Let me give you an example. You ever been asked a question, why are plants green? And the simple answer is, well, they absorb all the other light and they reflect green light. Yeah, that's not a wrong answer. It's also not very satisfying. I mean, why are they reflecting green light? And if you know anything about light, you know that green light has more energy than red light. So why would you absorb red light and reflect green light? I mean, wouldn't green light be better light? It's got more energy than red light. Wouldn't plant leaves be reddish? Or why not black? Why not absorb all light? So where we go from a superficial knowledge of something like, yeah, plants are green because they reflect green light, to really understanding why plants are green. Let's talk about this for a few minutes because I want to make sure you know the difference between superficial knowledge and true understanding, like a much deeper level understanding of the material. And here's a way to think about this. Okay, plants are green because they reflect green light. Not wrong, but not very good. Not very deep knowledge at all. I mean, anybody can recite that. So here's the example. Um, Plants are green, and the reason why, it has to do with the energy levels of electrons that form atoms. Atoms are made up of protons, neutrons, and electrons, and of course, the electrons are outside the nucleus, right? And they're in these electron shells and in different orbitals. And every time an electron moves further and further and further away from a nucleus of the atom, it gains energy. But an electron can only be in a specific energy state. It can't be in between. Okay, so red light and blue light, these are photons with energy moving in waves. Red light might have less energy than green light, but it has enough energy to elevate an electron into a specific energy level. Green light can't do that. Green light hits a plant, the electron goes up and goes right back down. It can't get it to a specific energy level. Blue light, on the other hand, can elevate electrons way high in energy, but it can put them once again in a specific energy level. And because these electrons must exist in specific or discrete energy states, green light can't do that. So the plants have evolved pigments that will reflect green light because they can't use it and absorb both red and blues on the spectrums. And that's kind of interesting to me. And when you learn even more about photosynthesis, you realize that plants are concentrating the energy by a process called inductive resonance, and it relies on these electrons going up in energy and then back down in energy and then transmitting it to the electrons next to them. It's like watching a wave at a giant football game, right? That's an example of inductive resonance. I always like it. Everybody goes, wave, and then the next person, they don't move positions, but your energy goes from you to them, right? And that's how inductive resonance works, to concentrate energy into the reaction center of the into the reaction center, where it elevates an electron really high in energy, and then that energy can be used in an electron transport chain to create a proton gradient, which through photophosphorylation also makes ATP, and through that we have now converted the energy in sunlight to chemical energy, ATP, that the cell can use, and it actually use that ATP in the Calvin cycle to fix carbohydrates, to fix carbon dioxide into an organic molecule. So these are the types of deep level understanding that I hope that you get out of this. Okay, so this week, like I said, study the photosynthesis. I am not going to have you memorize the Calvin cycle. I mean, I couldn't even have you do that on a test anyways. But you do need to know about rubisco, and you need to know about RUBP. Ribulose bisphosphate is the 5-carbon sugar that carbon dioxide gets stuck onto. And rubisco stands for, get a hold of this, ribulose bisphosphate carboxylase oxidase. I know, huge words. Ribose is a 5-carbon sugar, of course. Ribulose bisphosphate. I don't know why they don't just call it biphosphate, but it's got 2 phosphates on it. And rubisco is an enzyme that takes the carbon dioxide and sticks it on to ribose. And it's called a carboxylase because when you put carbon dioxide onto it, it of course forms a carboxyl group. So make sure you understand that. And then with the xylem and the phloem, I would definitely review transport from cell biology, both active transport, secondary transport, and make sure you know the difference between symporters and antiporters, and the difference between active and primary transport, and how they're used inside of plants, especially in the phloem, because they rely on both secondary and primary transport, and they're using both symporters and antiporters, which is, I think, very fascinating. Okay, so we'll go live this week. If anybody has any suggestions also for me, a way I could deliver this content to you better, please let me know. I'm open up for suggestions. I'm working on getting these really detailed PowerPoints. Remember that they're basically everything you need is in those PowerPoints. But I've also got a new podcast out on photosynthesis. I know that ever since Happy Gilmore, everybody goes, chlorophyll, that sounds like borophyll. I promise you, photosynthesis is anything but boring, and I tried not to get bogged down in the minutiae of exactly how the photosystems work or all the steps of the Calvin cycle. I'm less interested in that, as I am interested in the story of photosynthesis and how it has totally affected this planet, and how plants have adapted to dry environments by evolving both C4 pathways and CAM systems, and that's what that covers. Okay, until I go live this week, I'm going to try to go at some point, and I will send out an email to let you know when. Okay, have a nice week.